Cool. So today we'll talk about animal nutrition. So first things first, there are some essential nutrients that you should know. Um, for I wouldn't say you need to memorize these, but definitely know that these exist. So first we have essential amino acids, including isoleucine, leucine, lysine, blah blah blah. Okay, and then we have also essential fatty acids. So what these mean is basically you can't live without these because your body cannot produce these things. You have to eat things that contain these amino acids and these fatty acids. Um, so they're essential. And then here are vitamins. And if you want to be a real usable nerd, you should remember these. And notably, you should know niacin, so B3. Honestly, B1, B2, B3 you should know. B9, B12, you should also know. And then vitamin C, obviously. Sorbic acid, you should probably know that. And then um what I mean by remembering is basically your functions in the body. So B3 is in NAD plus NDP, and then also deficiency. So B1 is beriberi, um, and then it's all these things, and then like B3, you don't really need to know that, but you need to know that's NAD plus NDP. Scurvy is caused by deficiency of vitamin C. Cobalamin, B12, all these things. Vitamin A, retinol, and it was used to kind of treat blindness. Eat all So like vitamin A is in carrots, and then carrots are good for your eyes, so blindness. And then also vitamin D is used for rickets. Deficiency causes rickets, etc. And basically, what I say, what I remember is you know it's B1, but you also know its name in parentheses. So thiamine, riboflavin, niacin, like you need to know it. They use these names on and off. So sometimes they will say cobalamin instead of B12 or niacin instead of B3. So you should know when they use these words what these means. Also remember which one is water soluble, which one's fat soluble. But that should be really easy because if you can see fat soluble is only four vitamins. So it shouldn't be that hard. All right, so minerals. So basically, you need these minerals and a very small amount, and then these minerals aren't as important as the vitamins. So what you really need to know is well, these are not really, you don't really memorize them because you'll come across these in other scenarios that you would know. So I wouldn't take too much stress to memorize these. But obviously, you need iron in your diet. You need fluorine for your teeth. You need calcium, like that's basically for everything. Phosphorus, sulfur, potassium. These are not. These are required for you to live, but in very small amounts. So, like you see, like more than 200 milligrams. That's basically nothing. You guys probably drink more caffeine than that. Um. Okay, so food processing. So not like food processing making bacon, but food processing as in inhaling food. So basically, you have three, four, five different ways of inhaling food, but humans are actually bulk feeders, so they eat a very large amount of food at one time. And I cannot see the speaker notes for some reason. Um, that's too bad. But anyways, um, so basically, you have filter feeders, which are usually like humpback whales. They sit in the water, open their mouths, go swim up, and then they filter through all the stuff, getting all the plankton stuck in their things, and then eating that. Substrate is that you live in that thing you're eating. So like caterpillars, they're living on the leaves, and then they're eating the leaves and also pooping in the leaves. So they're substrate feeders. Fluid feeders, you're you're like drinking blood most of the time, so that's fluid feeding. And then bulk feeders are like snakes. Like think about like these are pretty like what they mean, also are what they mean. So like filtering, substrate, literally substrate, fluid, blood, and then bulk feeding, you're eating a lot. So it's pretty easy to remember. And then so food processing also involves like ingestion, digestion, absorption, elimination, and we'll talk more about that in next few slides, which is not the next slide. Um, okay, so First, digestive compartments. You have intracellular digestion, which is so here we have a hydra, and it is like a very like basic digestion system. So you have like a mouth, and then you have digestion. It gets absorbed into the body through the epidermis, and then it gets released presumably through the anus. And so basically, it get digested, and then all these particles, all these stuff you really need, are taken in by food vacuoles into your body, and then um that's how we digest it in intracellular digestion. But in extracellular digestion, you have like something called a gastrovascular cavity, or complete digestive tract, also called the alimentary canal. So that's basically what we have in the figure 41.7. So that you can see there are alimentary canals here. They're complete, and then that's basically what we humans have. I hope I didn't gaslight anyone. Mm, So yeah, so gastrovascular cavities are basically just one opening that, and for simple body plans, and then for the alimentary canal, that's like a complete digestive tract. So what we see in the grasshopper and the bird, and the earthworm is the alimentary canal. Cool. And then so the oral cavity, the pharynx, and the oh my god, did I forget how to pronounce this word? Anyways, um, it's esophagus. Let's forget that. Anyways, so basically humans, obviously. Oh, fun fact: the diagrams here are supposed to represent cadavers, so dead people. So fun to know. Um, so basically, oral cavity is the stuff in your mouth. When you open your mouth, it's also your opening oral cavity. It's, let me pause this. I can't see speaker notes whatsoever. Why is it black? There it is. Pharynx. No, esophagus. What? Is it spelled wrong? No, it's not. It's fake. Whatever. You don't remember what happened. It's Fogus. It's called Isfogus anyways. It's not that important. Hopefully, it's probably important. What would I know? Okay. Okay. So what is important is that saliva is secreted in the oral cavity. Wow. No way. Basically, so what's important about this is because saliva contains an enzyme called amylase, and amylase is really important in digestions of starch and glycogen. It's focus. Yeah. So basically amylase really important. But scientists don't know why, because not a lot of digestion happens in the oral cavity by the saliva. So, but the guess by scientists is that the food particles get stuck between your teeth, but the saliva helps break it down, so it doesn't cause cavities and stuff that you don't really want, and your dentist will get mad at you. Um, and then so basically you can see in this diagram here, see the tongue and the pharynx and stuff like that. So the tongue makes the food into create some sort of bolus of food, and then that goes down into esophagus. How do you pronounce it? This is bothering me too much. Esophagus. That's right. I need that. Esophagus. Anyways, so amylase, remember that, and then so it goes down. The food becomes the bolus, and it goes into the pharynx, which is the throat, and into the esophagus. Anyways, oh wait, no, actually no, it's yeah, and then the larynx and trachea. All this diagram is super useful. So I would say notes are very useful, but also diagrams. So a really good study tip, especially like 10 hours before the usable open exam, is to not look at your notes. Obviously, looking at your notes is important, but look at the diagrams because the diagrams tells you like a better picture, and then I think you're better off looking only at the diagrams than your notes. So you can see here, like this diagram shows your food going from your oral cavity into your stomach, which we'll talk about next. Um, but yeah, here and then um yeah, is there anything else important I need to say? Alright, the sphincter, which is kind of like a very reused term because you'll see this again in other parts of digestion. But sphincter basically is some sort of thing that constricts and then unconstricts to make stuff go through it, especially in your digestive system. Cool, stomach. Oh, my favorite subject. Anyways, oh, I forgot to say peristalsis. That's how your food goes from your throat into your stomach. So that's like the contracting of smooth muscles that gets your food to where it needs to go. Anyways, so stomach, first of all, the stomach has two uses. So first use is for storage. So you can store a lot of food. It can expand to around 2 liters. Depends on the person. Some people have more, some people have less. Um, but like a lot of fast surgery, like people who are like obese, they get surgery. They use like cut off a piece of the stomach, which is oversimplified probably, but that's basically what happens. Because the primary reason for the stomach is to store food, and the other is to digest food. So it primarily does this through gastric juice, which is also chemical digestion. So what we call this gastric juice and food mixture is called the chyme. And so what happens is basically food goes in your stomach. It sits there because you're not gonna start just digesting right away. Basically, your stomach it's really acidic, if you didn't know. Um, but it's around pH of 2 and mostly composed of HCl, hydrogen chloride, which is an acid, if you didn't know. Anyways, um, so this pH allows the gastric juice to denature proteins and expose peptide bonds. And these are attacked by pepsin. Pepsin is something important you should probably know. And then pepsin, the production of pepsin is also really important. So the chief cells produce pepsin. So they first produce pepsinogen and they get activated by hydrogen chloride into pepsin. And once they're done, it goes back. Once pepsin is created, it will kind of positively regulates this pepsinogen to pepsin. And then so the parietal cell produces the hydrogen ion and the chloride ion, and those become acids. Um, so yeah, the ATP pump drives out the H plus ions into lumen, and then the chloride ions diffuse into lumen, creating this acidity. Um, cool. I think I talked about everything. Wow, small intestine. So the duodenum is really important. So duodenum is just basically a small intestine, and it's the first 25 centimeters. And the chyme mixes with digestive juices. They release a secretin, and that stimulates the pancreas to secrete biocarbonate. And this neutralizes the acidity of the chyme and creates a buffer for the chemical digestion. Um, so, a primary thing method of digestion for small intestine and duodenum is absorption. So what we can see here is the carbohydrate digestion, protein digestion, nucleic acid digestion, fat digestion. So these are very different pathways because these are basically 4 different types of energy. So first you have carbohydrates, which is typically what you eat and what you get energy from. So that's a very good diagram showing like polysaccharides or disaccharides, how they get digestion. Um, so you can see if only for polysaccharides does it get digested in the oral cavity, into smaller polysaccharides and maltose. And maltose, you can see it gets digested in the small intestine, becomes polysaccharides, while um, enzymes from the intestinal epithelium, while the others get digested in the enzymes from the pancreas. Next, you have proteins. So proteins, they get broken down by pepsin into small polypeptides, blah blah. You should all I think for open exam cases to know that these are digested in different parts of the body, namely, the protein starts in stomach, nucleic acid starts in small intestine, fat starts in small intestine. And now you don't really necessarily need to know how each are digested, but you need to know like there are different avenues of digestion. Um, anyways, am I missing anything? Oh yeah, and then so the small intestine has something called the villi, and the villi has microvilli. So these are things that protrude out of the cell, and it makes the cell membrane, whatever the stuff you learn in AP Bio. I hope you guys are taking it and already know the answer. Basically, these projections increase surface area, hence absorption is greater. Pretty logical. Okay, so the large intestine is the colon, the cecum, appendix, and feces. Feces is the poop. We all know that. And then, so the colon is what it starts off with. So, let's see. So colon leads to the rectum and anus. So anus is where all the stuff comes out, lack of a better term. And then I say the cecum is used to ferment the ingested materials. I think there was a really good diagram, but I didn't make these slides. So, um, let me try to find it. But it was a really good diagram. But basically, the CISA is very important because like if you're a herbivore, like the cell walls are really hard to digest by its own. So basically, this for herbivores, the large intestine and the small intestine are relatively the large intestine, I would say, not the small intestine, but the large intestine is relatively longer, especially the cecum are longer than omnivores or carnivores because they need to break down. There's so many cell walls. They need this fermentation process to get nutrients, and so that's something you need to know. Herbivores have a longer digestive system, namely a longer large intestine than omnivores or carnivores. And the carnivores has a very short digestive system because all the protein, um, all the fats, they don't need to get digested. They don't need to be fermented, namely in the large intestine. And the appendix, it's like a reservoir for symbiotic microorganism, but like it doesn't serve a really big purpose, especially in humans. But it serves a big enough purpose, so if you get rid of it, like they would usually get like a long time ago, decades ago, they would usually get rid of it if you get a C-section when you're giving birth, and then that will cause a lot of health problems, and you're like, whoa, what's going on, man? And they stop doing it. So unless it's in danger of bursting, which is endangering your life, they don't usually remove it. And then finally, feces. That's the poop, and then it gets stored in the rectum. All right, evolution adaptations. So here you can see there's incisors, the canines, the premolars, and the molars. So you can see here the carnivores have more they have canines, large canines, and in herbivores they have a lot more molars, and omnivores they have kind of a blend of both. So they kind of make sense because carnivores need to tear apart meat, herbivores need to chew these plant stuff, and then omnivores need to do both. Oh, there was this picture. I was thinking about it, but so you can see here the cecum is like super long. Large intestine is also super long, and the small intestine is relatively the same, but the large intestine is definitely much longer. Oh, I don't know what the slide is for. Um. Oh, microbiome, mutualistic adaptations. So the microbiome is the stuff, bacteria, and then living on your body, in and out of your body that helps you, protects you, keeps you healthy and stuff like that. And these are mutualist adaptations, and there's a ton of bacteria. So I hope you all know that. Anyways, I think we skipped a few things, but did we? Am I geeking? Definitely skipped a few things, especially insulin and glucogen. But I'll just go over it quickly. So basically, we this chapter also has diabetes, which I don't know why it's not here. Anyways, so diabetes is basically you can regulate your blood sugar, and the diabetes mellitus has definitions deficiency of insulin or decreased response to insulin. And then so basically the pancreas with all of the alpha and beta cells, they're not working. So alpha cells produce glucagon, and then the beta cells produce insulin. And then insulin helps, lower the blood sugar, while the glucagon raises it. And then I won't go to details because these are all in campbells, but you need to know what I'm telling you right now. And so type 1 diabetes is basically insulin dependent diabetes. So basically your like immune system kills all your beta cells, and then you physically cannot produce any insulin. So that's type 1. And type 2 is non-insulin dependent. So basically you still produce insulin, you're still producing glucagon, but your body does not respond to it really well. So type 2 is typically acquired later down in life, while type 1 you're born into it. So that's the difference. And then chapter 42, circulation gas exchange. This one's pretty difficult. If you're in science school, you should probably know this. Anyways, um, okay, so I'll just jump right into the really important bit. So here you can see in the bottom, this is a diagram of 3 different animals. So you first have fishes, amphibians, and mammals. So double circulation will be like reptiles, amphibians, and then uh, here you can see. Okay, I'll just go through. I'm going too fast. Anyways, so first fishes. You can see here the capillaries. So capillaries are like capillary beds, we call them. So there's like really, I can't even call them blood vessels, but really thin blood vessels that are typically like lungs or your legs, stuff like that. So here it's gills and body. So really simplified diagram. And then so the gills get oxygen from the water, so it becomes red with all the oxygen rich, and it goes into the heart. So the heart, you can see there's only 2 chambers. So you can see atrium, 1 chamber, and then ventricle, the 2nd chamber. So basically, the vein takes blood into the heart, while the artery takes the blood out of the heart. Um, And then veins have valves, arteries do not have valves. The veins are typically lower in blood pressure, arteries are higher in blood pressure. So remember that. Um, so remember, fishes have only 2 chambers in the heart, while amphibians and reptiles, they have 3. And then not birds, specifically not birds, because birds are typically classed together with reptiles, but birds actually have the same circulation as mammals. Um, so here in amphibians and reptiles, you can see that they have 1, 2, 3 chambers, so 2 atriums on 1 ventricle that is mixing. The right and left right atrium, you can tell like the right and then the left are not your right and your left. And this is because, as I said before, in these diagrams we're talking about like the heart, stuff like that, they're literally using cadavers to learning about this. So it's literally the cadavers right and left. Um, so that's why it's like this. Um, so the atrium, okay, the vein takes the blood in the atrium, and this is oxygen poor blood, and it goes into the lungs or the skin, which is how amphibians breathe, and then it goes back into the heart to the atrium again, and then the ventricle pumps it out into the other capillaries. And then finally, you have mammals and birds, which is 1, 2, 3, 4 chambers of the heart. And we'll dig into deeper about specifically what each chamber does, but for this diagram, it's really good. So Fish, 2 chambers. Reptiles, 3. Mammals, 4. Okay, and then they test this all the time in open exam and science bowl especially. So anyways, and they also have an open and closed circular system that I probably should mention. So open circular system is something like a cricket will have, a grasshopper, an insect will have. So basically, you have a heart, but these it's we call it hemolymph. I think Anthony talked about it last week. Well, last week, but hemolymph is what we call it in all open circulatory systems, and then so basically the heart pumps it into these cavities, in the body that like um gastrovascular cavities. I think is it. Let me double check. I think it is. Well, anyways, it's cavities that kind of bathe these organs and muscles with these hemolymph with the oxygen. While in closed circular system is what we have. So these are containing vessels, and we call them blood. Cool. Wow, such a good diagram. Honestly, you can probably learn Campbell's using only the diagrams. Anyways, so here you can see a more generic, comprehensive graph of diagram. So here we have a mammal heart. So you can see 4 chambers. So it goes, the vein takes in all the blood, pour oxygen, pour blood into the right atrium, and the right atrium leads to the right ventricle, and the right ventricle goes to the heart through the pulmonary artery, and pulmonary artery goes to the lungs, and then comes back into the left atrium. Left atrium. Oh, I think I messed up the order, but it's still the same order, right? Anyways, I just did it in the wrong starting point, but whatever. Um, Goes into the left atrium. Left atrium goes to the left ventricle. So you can see it's red, so it's oxygen rich, and then it goes to your body. Comes back, oxygen poor. So the aorta takes the blood out of the left ventricle. So it's a lot of stuff, but you definitely need to know the order. So the order typically will be like body. So our body to they'll probably say like right atrium, right ventricle, and then they'll talk about pulmonary artery, and then you have to say the order, correct order in the usable open exam. So definitely remember all these words and how the blood flows through, and remember to not confuse yourself with right and left. Think about the fact they're looking at other humans, and that is their right and left, not yours, right? You can always imagine yourself, but I wouldn't think you would want to imagine yourself being dissected. So that's how it works. And then so also I want to mention the SA and AV node. Um, anyway, so here you can see the semi-lunar valve and a AV valve here. You don't really need to know this for the open exam, but you probably do need to know this for the semis. Um, so open exam, you only need to know the order generally. These for open exam, the general rule of thumb is you need to know how everything works, not any specifics. So here in the cardiac cycle, you usually have a systole and a diastole phase. So systole is the contraction, diastole is the relaxation. And when you're getting your blood pressure measured, there's a systole pressure, and a diastole pressure. So these are measuring typically a contraction and a relaxation. The AV valve prevents the valves, as I mentioned before, they prevent backflow, and then sometimes they don't really work. So that causes heart murmurs, and then if they don't work, that's really bad. So you need open heart surgery. Anyways, so the SA node is important because like, that is your pacemaker of the heart. So that determines how fast your heart is beating. And um I don't know if Derek's nickname is still this, but his nickname in Discord was per Purkinje Fibers. But that's also really important, but not for the open exam. But all you need to know is the SA node is the pacemaker. and AV node is a relay point that relays it to the Purkinje fibers. Okay, as I was talking about, valves have um, veins have valves, arteries do not have valves. So this is a very good way. So veins have started with a V and valves started with a V. So you remember which one has valves, which one does not. So here you can see an artery and a vein. So the artery here you have a smaller vessel because of higher blood pressure, smooth muscle to move it along. And then here in the vein you have the valve, less smooth muscle, and it's like generally less thinner walls because it's not very high pressure. Um, here you see sphincters again, they just contract flow. So sphincters contract and relax. That's a really general term. So vasoconstriction is when your blood vessels are constricted to increase blood pressure, while vasodilation is the opposite. We also have a lymphatic system that accompanies our cardiovascular system. So lymphatic system is like a completely different system, and it basically works together. And then like if you have a cut or wound, all the cells come in through both the blood and the lymph. Um, so that's pretty much what I think you need to know for the lymph nodes. So yeah, lymph is basically like white blood cells and your body's defense basically. And and I think we should also know there's an organ called a spleen. And I don't know, in Capos is briefly mention it, but I think in usable, open, and especially semis, they talk a lot more about it. So the spleen, you should probably know about it, know about its existence, but I wouldn't say you need to memorize what it does. So basically, the spleen, what it does is stores and filters the blood and makes and creates the white blood cells. Anyway, so stuff you have in the blood, you have stem cells. I hope you know that because everyone researches it anyways. This was stuff we'll talk about in the immune system, I think today too. I don't know how I'm gonna get to it. But so in the blood you have the plasma, erythrocytes, leukocytes, and platelets. Erythrocytes is red blood cells, leukocytes, the white blood cells, platelets, and the LDL and HDL. And it basically all these kind of play a role in immune system, so we'll talk more about it later. Um yeah, I don't want to talk about it right now. So okay, gas exchange. Wow. Okay. Gas exchange. So you have surfaces in your body. Each organism has surfaces, epithelium that works in gas exchange. And oh my god. Anyway, so here you have a picture of a lung, and then you can see here the nasal cavity and the larynx, the esophagus, trachea, and then it goes into your lungs, and the bronchia, bronchial, whatever. So lungs, you know how it works. And then in the lungs, you have alveoli, which is like the endpoints, and then that allows your blood to from oxygen poor to oxygen rich. You also have gills, so like in crayfish, and sea, so anything that lives in the ocean, they have gills, and then basically need it to get oxygen from the water. No way, anyways. Also have you have something called counter current exchange. So this is the exchange of a substance between two fluids flowing opposite directions. So that happens especially in gills, how gills work. So that's counter current exchange. So the blood flows one way and the water flows the other way. I think there's a better diagram somewhere. Anyways, partial pressure, I hope you guys know what that is. So partial pressure is basically like if I want to take the partial pressure of oxygen, O2, then like the total pressure will be something different. But partial pressure is that if I remove every other gas and only had oxygen left, what will be the pressure? So that's partial pressure. So breathing. Anyway, so breathing ventilates the lungs. So what happens in the human body is that your diaphragm, it pulls down for inhaling. That creates kind of a negative pressure, so it fills that gap, the lungs, to breathe, and then it squeezes out that air in your lungs. So that's negative pressure breathing. But um, birds in part, I would say, remember how birds breathe, because birds are like, there's typically like positive pressure breathing, which is amphibians, um, inflating lungs with forced airflow. Um, but I would say birds are probably more tested upon, because birds are they live in high altitudes because they fly, and then oxygen is not a lot in those high altitudes. So they have this breathing system that's way more efficient than positive or negative breathing. That I can't really go in detail right now because I'm short on time. But just know that birds have a separate system. Birds are like they're different from reptiles, like mammals, but also different from mammals as well. Anyway, so we have something called myoglobin plus hemoglobin. Hemoglobin is your prime. It's made out of iron. Um, so hemoglobin, you have four subunits. Each subunit can carry one O2 molecule. And honestly, like for juniors, I don't know if there's any juniors, but you'll learn about a lot about this in inorganic chemistry, because that's the iron metal complex, but you don't need to know that. And so we have hemoglobin, but also on the side here you have myoglobin, and myoglobin is typically used in your muscles. So when you see like a steak and it looks very rare, and then like the pink stuff starts flowing out, and that's myoglobin, not blood. So it's very different, but also does the same thing, which is provides oxygen for the muscle, for myoglobin, but hemoglobin for all the body. Mm, what else I have to talk about? Oh, also bore shift. Bore shift is really important. Bore shift is like if um I did not review this beforehand if it wasn't obvious, but bore shift is like in certain conditions, if it's saturated, then you can see here, because if there's carbon dioxide, hemoglobin will bind to that rather than oxygen. So you want something that has a lot of oxygen in the environment, so you can breathe and not breathe in CO2, because that actively competes with oxygen. And then, but there's also other things you can search up bore shift or in Campbell's. I'm pretty sure it's in there, but Campbell doesn't really talk a lot about it. I think Wikipedia does a better job, but definitely remember how bore shift works. Anyways, immune system. Man, 45 minutes already. Dang. Okay, so basics of immune system. So immune system is comprised of innate and adaptive immunity. So innate is kind of like it recognizes traits shared by a broad range of pathogens. Then so you have barrier defenses, so skin, and then internal defenses such as natural killer cells, NK cells, blah blah blah. And then adaptive immunity is only seen in vertebrates. Vertebrates have both innate and adaptive. Adaptive is they recognize these things, and they specifically um, have things that attack these pathogens. So they produce more of a response to antibodies and cell-mediated response with cytotoxic cells. Um, Anyway, so innate immunity in invertebrates, so they have exoskeleton that is a physical barrier against infection, and then chitin makes up that exoskeleton. Lysozyme breaks down bacterial cell walls if they do get infected, and also they have hemocytes, so they basically ingest and break down microorganisms by phagocytosis. Um, yeah, recognize antigens and nom-nom. And also have toll receptors. And we'll talk more about this when we talk about vertebrates, because they have toll-like receptors. But toll receptors, they coordinate release of specific peptides in order for defense. And in mammal, you can see here, mammal innate immunity. So you have same barrier defenses, but this time you have skin and mucous membranes. Also have lysozymes, but these are located in the tears and saliva and mucus in your body. And then you also have toll-like receptors, so they bind to epitopes specific to the pathogens. And you have eosinophils. They target multicellular pathogens, natural killer cells to release chemical that kill cells. So you need to know natural killer cells, dendritic cells, macrophages, neutrophils, eosinophils, dendritic. Like these are all innate immunity. So they typically I want like it's seen in opening exam. Like they ask you like, okay, what is immunity? What is active in the immunity? And they give you like a list of cells, and you need to recognize all of them, right? So definitely remember that these are innate immunity. They don't rely on targeting. They have like a really broad range, and they attack everything they see. Um, and then also we have local inflammatory responses. So here you have okay, so you have a splinter. The splinter goes into your body and it leaves a hole, and then on that splinter has a lot of pathogen. So here, once it detects that, your blood cells, it capillaries widen, and then the neutrophils, everything, mast cells, macrophages, they all come and they release signaling molecules for the neutrophils to swallow up all the pathogens. Nom nom. So mast cells release histamines. Histamine is also amino acid, so remember that. And then causes capillaries to widen and allows for more fluids to go in to heal that splinter. Cytokines also release cytokines recruits neutrophils. Neutrophils also go and swallow nom nom, and this causes a positive feedback loop. Okay, so fever happens to fight stuff, and then like this is pretty logical. I hope I don't have to explain to you, but yeah. So basically, you want to increase a lot of the production of the stuff that will fight against whatever's infecting your body. And also, you can go into septic shock, which is high fever, low blood pressure, and poor blood flow is commonly seen in elderly and infants. Um. Okay, so interferons, they interfere with viral replication. So infective cells, they secrete this interferon, and they also inhibit viral replication. This can also be produced by white blood cells. And then yeah, they increase their phagocytic abilities. Let's see if I can get through this. Adaptive immunity, cool. Adaptive immunity is B and T cells, so lymphocytes, and they're produced in the blood marrow. So inside your bone marrow, what is going on? Bone marrow. B cells, they mature in the marrow, and T cells, they mature in the thymus. Um, Thymus is, I don't remember where it is located because I washed anyways. Um, So the B cells they remain and mature in the blood vessel and blood bone marrow. Antigens they elicits the B and T cell response. Antigen receptors include epitopes, so there's small accessible portions of antigens that bind to an antigen receptor. So B cells. Um, Whoa, okay, I think we jumped a lot. Anyway, so B cells they have antigen receptors. So as you can see here, it's a Y shape. There's an antigen receptor, and they also have B cell. How they activate it are they first bind to an antigen, and then it replicates because it recognizes the antigen, and then it clones to make antibodies, including immunoglobulins. So immunoglobulins you need to know IgA, IgD, IgE, and IgG and IgM. So what you need to know is that there are these types, and specifically you need to know two. So IgE, well, I would say IgG is the most important. You need to know IgG. Like there's no excuse for not knowing IgG. IgG immunoglobulin G is like 70% of all immunoglobulins in your body, and they activate the classical pathway of the complement system. And the most important part is that it can cross the placenta and it gives passive immunity to a fetus. So that's the most important part. It's the most common immunoglobulin, but also can go into the placenta. Okay, and the IgE is the one that underlies your allergies. So it triggers histamine release from mast cells and basophils, and that causes your allergy response. So you need to know these two functions, but you also need to know the IgA, IgD, IgE, IgG, and IgM. Okay, T cells. Um, so you have a T cell antigen receptor, and then how it activates that receptor binds antigen fragment presented by MHC molecule. So you can see here, MHC molecule presents this antigen to that T cell. So it has the pathogen, and the fragments break off, and it presents that lymphocyte development. Okay, so this is causes basically B and T cells. You know, they're stuff relying your immune system, immune response, but they need to have diversity in order because they're adaptive. They need to adapt to the scenario, and basically they need diversity. Um, I think we can skip this because it's not that important. Anyway, so that's what I was talking about. So adaptive immunity activation. So antigens presented to a stream of lymphocytes in the lymph node, and when a match is made, this specific lymphocyte proliferates. They form memory cells, but also effector cells. The memory cells, they stay in your body for like a certain period of time, and if the same thing comes and affects your body again, the memory cells then proliferate. So it was a faster immune response. So that's the immunological memory. And there's a primary and secondary immune response. Mm, So primary, they peak around 10 to 17 days after initial exposure, while secondary is 2 to 7 days after exposure. So primary is like when it happens, first happens, but secondary is when it already happens, when you already have those memory cells. And then so T and B memory cells to be exact. Um yeah, adaptive immunity defends against infection of body fluids and cells. So helper T cell, I don't know who added that. Anyways. So helper T cell binds to antigen presenting MHC molecule, and the binding results in cytokine release from both the T cell and represent T cell, and the result proliferation of that T cell. And T cell clones secrete more cytokines and activate both B and T cells with the same receptors. So that causes the adaptive immunity. And then this they cut off the description, but whatever. So basically here, antigen presenting cell engulfs a pathogen, degrades and displays the antigen fragments. Um, the class 2 MHC molecules on cell surface, and then the helper T cell binds this and via the antigen receptor and the protein called CD4. So here, and then the binding causes secretion of cytokines, and then these cytokines will activate hyper helper T cells and stimulate its proliferation. And here, cell proliferation produces a clone, and then you have B cells and cytotoxic T cells. B cells are humoral immunity, and then T cells, cytotoxic T cells, are cell-mediated immunity. So they attack affected cells, while humoral is secretion of antibodies. Whew, there's more. So essentially, what you have here. So B cell activation, B cells bind to antigen, present only that antigen to its MHC molecules. Activated helper T cells binds to B cell. MHC releases cytokines that activate the B cell, and the B cell proliferates into plasma cells. And then these secrete antibodies. So here you can see here something kind of similar. So you still have the class 2 MHC molecule, and you still have the antigen receptor. So it engulfs the pathogen, the antigen presenting cell. It displays the antigen molecule. Helper T cell recognizes this, and then with cytokines they secreted from the antigen presenting cell. Here you can see that process going on. And then B cell with receptors of the same epitope internalizes the antigen and displays the antigen fragment on the cell surface with the class 2 MHC molecule and activated helper T cell, which has the receptors specific for display fragments, binds to and activates the B cell. And an activated B cell then proliferates and differentiates into memory B cells and antibody secreting plasma cells. And the secreted antibodies are specific to the same antigens that initiate the response. Whew, a lot of things. Immune system is not fun, guys. So what exactly do antibodies do? So antibodies, they interfere with pathogen activity and mark pathogens for inactivation and destruction. They also neutralize them, so antibodies bind to proteins on the surface of a virus and prevent the infection of a host cell. And it also do optimize, optimization, and antibodies bound to antigens present these recognized structure for macrophages and neutrophils, and these promote phagocytosis, so they swallow these macrophages as seen here. Opsonization. And then yeah, natural killer cells also recognizes these to do what the name says, natural kill these cells. Okay, cytotoxic T cells, they respond to infected host cells. So they use toxic proteins to kill cells infected by viruses or other intracellular pathogens before these pathogens fully mature. So they require signals from helper T cells and interaction with antigen presenting cells to activate. And this before we talk about like CD4, but now it's in cytotoxic T cells, it's CD8 proteins with the class 1 MHC molecules. So here, activated cytotoxic T cell binds to class 1 MHC antigen fragment on an affected cell, and then T cell releases the peripherin molecules and they form pores in the affected cell membrane, and then enzymes break down these proteins and enter the cell, and then it dies. I don't know why the title is this, but so basically vaccines. I hope you guys know what it is. And an active and passive immunity. Mm, are you? I don't know why you skipped this diagram, but it's a really good diagram, Campos. I hope you guys will look at it because that kind of generalizes what's going on. Starts off with like antigens, going to antigen presenting cells, but and what happens exactly. And I think it's a very helpful diagram. Um, nope, it's if. Never mind. Never mind. Anyways, so you have vaccines and active and passive immunity. Um, so active immunity, they defenses that arise from the pathogen, infection, or immunization prompts an immune response, while passive is antibodies and recipients produced by another individual. And then antibodies as tools, you have monoclonal antibodies. So they're produced by a single clone B cell, and they're useful for tests. Also, immune rejection and blood types. So A, B, and O. There's also different blood types, but for open exam, you only need to know A, B, and O. But for semis, you need to know all the different blood types. Cool. And then, God. Okay, so you have allergies, as I said before, caused by IgE antibodies. So allergies, these when exposure release B cells to make IgE, and these bind to mast cells, and mast cells release histamines, and you go into anaphylactic shock. Well, not necessarily, but anaphylactic shock is what happens when the allergies get so severe, and then you need EpiPens to rejuvenate yourself. Am I going overtime? Oh, great. Anyways, and some evade the immune system, so you have AIDS, so they like keep silent the entire time, and they don't get recognized, and that causes AIDS. So HIV, that virus causes AIDS, and then basically when you get it, you're done. And latency, they stop producing, and then proteins become invisible. Cancers could be caused by viruses and antigen variation. So that's when you get a flu shot every single year because they mutate a lot, and your body cannot handle it. Why is this slide here? Someone move this. Oh, this was the stuff I was talking about. This is the diagram. So let's stick for 1 more minute. So basically here, antigen taken in by antigen-presenting cells or by B cells. Helper T cells produce more B cells and cytotoxic T cells. You can see here, this diagram basically summarizes what I told you for like 30 minutes. It's very helpful. I say when an open exam happens, definitely look at the diagrams rather than your notes because cramming your notes is probably not the greatest idea.